in a group called Computational Science in Amsterdam. And this is a very broad group, so we are interested in building mathematical models and running simulations in many different fields. So not just the biomedical field, we also have projects where we look at other types of systems in sociology, psychology, but also in physics and chemistry. There are lots of collaborations with applied math groups, with groups that come from various fields. We are quite active in the complex systems community, where we develop theories. But for now, my main focus will be on multi-scale modeling in cardiovascular disease, on biomedical applications. Well, I didn't know what to expect, and I thought I will take a long introduction, but maybe if this is all known to you, please let me know, and I will jump in the real topic of the talk with. I'm also affiliated to the University in St. Petersburg, so we have some long-running links between IPMO and University of Amsterdam. So we have lots of exchange of staff and students between both universities. And that's something that we find very interesting to develop also with Russian institutes here and with other institutes here in Moscow or in other cities. We had a workshop on computational biomedicine about 1.5 months ago, where we invited some scientists mainly from Moscow and from St. Petersburg, but also from Nizhny Novgorod and some other cities. And it was, for me, extremely interesting. We liked it a lot because there were many new contexts coming from that. Okay. I want to first discuss with you the virtual physiological human. This is an initiative developed over the last 5 years in Europe, mainly paid by the European Union. And the main idea is that what we want to do is to model human physiology. So to create multi-scale mathematical models of human physiology that should be descriptive, so we should be able to basically put physiology in a computer in order to better understand it. It should be integrative in the sense that it's not just about a very specific part of the physiology that we want to understand, but in its context of the rest of the human body. But most important, it should be predictive. So we want to be able to predict certain features of health and disease. Okay? But for most of you, that's pretty straightforward, so I'll quickly move on. So it's basically, and these are, by the way, slides from the Virtual Physiological Human Initiative, so it's a bit of a, let's call it an elevator pitch. So don't make too much out of this. But it is important. So far, we have seen deterministic science, trying to understand all the little bits and pieces of the human body, and now we have gotten to a point that we want to put the pieces back together again. That's basically the metaphor that we want to use. Don't read the text. So we want to model how the human body works, and you can do that on many different levels. And the whole idea is to pick the right level where you want to study it, and then connect. For instance, if you're interested to study tissues, again, it's all about mathematical modeling, to also include models on the organ level or on the cell levels and all that. So we want to create multi-scale models in biomedicine, and this should be across temporal and dimensional scales. So it's going from, if you want, from molecule to man, that would be this thing here, or even to a single human being in a population interacting with other human beings, or in the environment. Typically what we do is we pick a certain level on which we study, and then we bring in functionality. Well, this also means, and this is where mathematical modeling is very strong, it also means integration across disciplines. That's bringing together numerical mathematics or applied mathematics with medicine, with biology, but also with physics and chemistry and all that. Important is also, and that's something we do, we also bring computational and computer science. If you want to build these kinds of integrative models, it's all about being able to integrate these models. Okay, now, there is a thing called the VPH Institute, so if you're interested in these activities in Europe, this is the place to go. So the VPH Institute.org. It's basically a community effort. So many groups in Europe that have been working on projects like this decided that if we want to stick together as a community and we want our models to further develop, we want to share these models, we need something. And that something is the VPH Institute, and this is slowly becoming a worldwide initiative. I think it's very interesting. Now, there are two specific applications that I wanted to discuss today, and I will basically focus only on this one here as an example of the kind of modeling that we do and where I believe this could be going. And we are interested in one project in the treatment of intracranial aneurysms. Well, that's the one I won't speak about, but if you're interested in this, let me know. I can share, of course, what's more of this with you. The other one is an example of coronary artery disease and treatment of coronary artery disease. And I picked that one because it is an interesting story to tell. We have many nice results on that. So I will explain what instant restenosis is and all that. So maybe a few statistics. Most of you are aware of this. Cardiovascular disease in Europe and also here in Russia are the most common cause of death. And it's approximately 4 million deaths each year. It's just basically half of the total amount of deaths per year. And this is a statistic from 2 years ago. Well, half of this is due to coronary heart disease, and 1/3 is due to stroke. And so brain infarcts. Well, of course, one big problem is, and this is something we see a lot in all these diseases, is stenosis of coronary arteries. And so the coronary artery is the artery that provides the heart muscle with blood, and stenosis is a narrowing of the artery, basically meaning that you get less supply of blood. This is, of course, due to bad eating habits, too little physical exercise. Well, this is by now the standard treatment in the cath lab. So you go to the hospital. Basically, you can be treated. It's almost, you stay overnight in the hospital and then you're out, in principle, and everything goes well. However, this is a very successful treatment and it's applied a lot. There are many people who receive this balloon angioplasty and stenting. However, something can go wrong, and this is called restenosis, that for some reason scar tissue develops in the artery. And this is called instant restenosis, and this happens in 5 to 10% of patients. Well, given the sheer numbers, because this treatment is done so much, 5 to 10% is really a huge number of individuals. And if it happens, it may lead to a next treatment, again a balloon angioplasty, or even open heart surgery if nothing else is possible. Okay, so I want to talk to you a little bit about how this looks like. So if you would take an angiogram, that's this thing here, the white parts here are the coronary arteries, and if you look closely, well, it's not good to see, but here, there is a severe stenosis. The arteries are blocked. And, well, if you would take a cross-section of such an artery, it could look like this. And so this would be the normal lumen, the part where the blood would flow, but it's completely blocked by that. This is an extreme case, of course, but just to make a picture. And here you see a very small part of the blood will flow. Okay. This is then how the treatment looks like. So the medical specialist goes in with a guide wire. A little balloon is inflated with water pressure up to 10 bar to really crack open this stenosis. And then if that was all you would do, you would remove the balloon. In more than half of the cases, the artery cannot stay open, it will just collapse. So you need a scaffold to keep the artery open, and that's the stent. That's this little iron thing here, and this is just an artist drawing, of course. And you keep this thing in the artery, and you keep the artery open. And, well, what happens is, of course, that because of this treatment, you have damaged the artery, you have damaged the internal tissues, and a biological healing response starts. And so if everything goes well, you develop a nice new endothelium and everything is perfect. However, this process can go out of control. And that is what we call restenosis. I will show you that later. Here you see a little picture of one of those stents that goes into the artery. So this is the tip of a finger, so that you get an idea of the size of these devices. So this is what you hope to see. Here you see a coronary artery before treatment. Now it's done with contrast fluid, so this is a CT scan. And this is how it would look like after treatment. So it's nice open, you can see the guide wire here. And this is what the medical doctors want to see. But it should stay open. And what can happen, of course, that if you look 6 months later, that actually you can go back to a situation like this, where there was so much scar tissue developing inside the artery that you have to start over. So this is how that could look like. So if you first concentrate on this picture, so now what you are looking at is this coronary artery, a stent is in place, and then a cross-section is made. So these black things here are basically the stent struts, as they are called. So these little wires that are in the stent. And what you see here is, if you look carefully, this is the original artery lumen. And some new tissue has formed here. That's this stuff here. And you have a nicely formed new lumen, if you want. And the blood can flow freely here. However, if you look at this example, again, you recognize the struts now, the original artery, but there is lots of tissue has formed here, and almost half of the area is covered by new tissue again. So this is called the restenosis. Yeah, and so you have seen the, I don't have to talk you through this again. So restenosis is this abnormal growth, the hyperplasia of new tissue, and you want to prevent this, obviously. Now, when we started to think about this a while back, this was in the era that, so like 6-7 years ago, that so-called jockey-luting stents came on the market. So what you can do is, of course, take a stent, put a little polymer on it with some drugs in there that prevent the growth of this tissue. And these jockey-luting stents were extremely successful. So basically, it seemed like the problem was gone. However, because these drugs prevent the growth of this tissue, they also prevent the actual healing response. So these jockey-luting stents still have a problem with what is called late thrombosis. So for some reason, they can develop blood clots after 6 months or 7 months that can form an emboly and you get a heart attack. So there are still problems associated with the current stents, which are jockey-luting stents, or even more advanced stents. And we said, well, we would like to bring all the knowledge, biological and physics knowledge that is available for this system, build a multi-scale model, just to first better understand what is actually going on in the system, so understand the disease, and if we understand it, can we then try to improve the treatment? And that was a program that we have started, and we have developed a series of models for this. So, to repeat what happens, you deploy a stent, you get a severe injury in the artery. So what for sure happens is that the complete layer of endothelial cells is gone inside. And so if you have an artery, the inside of the artery has a monolayer of endothelial cells, and these cells are extremely important in the physiology of an artery. However, you can also damage what is called the internal elastic lamina. So that's the next layer in the artery if you go from the inside out. And if you damage that, you go into the adventitious. It is stretched for sure, but it can also rupture. What very quickly happens is an inflammatory response followed by activation of the vascular smooth muscle cells. These vascular smooth muscle cells start to migrate, they start to move, and then they start to proliferate, meaning that they start to grow and to divide. They deposit extracellular matrix, and this activation of the vascular smooth muscle cells leads to this new tissue. And so we have built a model that mimics this behavior of the vascular smooth muscle cells. And we have built in quite simple rules to follow this process. Now, you can play this game, of course. In all these systems, you could try to model the biological processes. But you know, there is no use if you don't have real data. And we only started this because in Sheffield University, a cardiologist, Julian Gunn, had an enormous amount of samples of in vivo data of in-stent restenosis. And what they did is they took pigs, they stented the pigs, and then they watched the process develop over time of the in-stent restenosis. So there are like 500 plus samples of stented porcine pig arteries, and they collected this over a 10 years period. And there is an enormous amount of data. We are currently only using a subset of this data. And it looks like this.
They are open, this data.
Sorry?
They are open, this data.
It is not public domain. If you're interested in it, then we should talk. I'm sure Sheffield would allow access to the data. But it's not public domain. No. I think I talked to them about this because it would make sense to make the data available. But it's a huge. I mean, basically, this is what the data look like. And these are just sections of these arteries, and there's histology. And the next step is to actually work on these pictures to get some real numbers out of it. And we are still doing that. Good point. Julian Gunn defined the so-called injury score. And the idea, I'm not going to explain this injury score in detail, but the idea is as you push the stent deeper into the tissue, you create more damage to the tissue. And he had a scoring system from 0 to 4, and believe me, you know, it basically correlates nicely with how deep you push the stent into the tissue. And we have checked that, and that makes sense. Now, the kind of data we have is the following. So as a function of the injury score, which is on the horizontal axis, we measure, well, as a function of time. So each graph, this is 6 hours after stenting, 2 weeks after stenting, 3 or 4 weeks after stenting. We measure for each injury score the neointimal thickness, which is a measure of the in-stent restenosis. So every single dot here is basically measured on a single stent. And so that's basically each pig is stented on 4 or 5 different locations. So you can see that there are many animals who sacrifice to do this. It's quite a cumbersome process. But even, you know, if you look at this, you know, with say, if like myself, if you come from physics where you used to have lots of data and very precise measurements, and this is a very sparse data set for us to. I will show you later why. We decided to plot this in a little bit different way, namely having time here and the neointimal thickness here, and then each line represents an injury score. So basically what we see is, first of all, you know, we have only, you know, immediately after stenting we have some data, but that's not so much. Then only 2 weeks later, and then at 3 weeks and 4 weeks, and I would like to have much more data here. So we are currently developing mouse models. That's a little bit easier than with the pigs. Well, I mean, with this data, you notice a few things. First of all, if you look at the initial response, if you have more injury, you find a faster initial response, so a faster initial growth of the neointima, and that's basically what you see happening here. And then after 21 days, you find that it seems to silence, or well, this could also suggest that it goes a little bit down, but I would say after 3 weeks, basically the response stops. And there is a positive correlation between the amount of neointima and the injury score. That's what we learned from this data. And also the time frames in which this happens. So this is for pigs, so after 3 weeks, basically the response stops. In humans, it takes a little bit longer, up to 2-3 months, and then also the response stops. Now, we have developed a mathematical model for this, and we quickly realized it must be a multi-step model, in the sense that you are looking at processes that happen in a single cell. The vascular smooth muscle cells, they start to proliferate, they start to divide, but you are also looking at processes that happen at the subcellular level. I don't know, cell signaling between the cells, pathways that are switched on and off, but also on the level of the tissue. So this overall tissue is developing, blood is flowing, there is an interaction between the, let's say, the physical signals from the pulse of our blood flow and all kinds of pathways in the cells. So we quickly realized that this is a pretty complicated thing with many, many relevant processes, as I said, from the subcellular level to the tissue level. And they all are coupled. And it was not, at that point in time, not clear which process was the most important process. So basically what we did is we went in the literature, we tried to create a list of all the what we call single-scale models, biological single-scale models that are important. And with single-scale, I mean, is this a model that is relevant on the molecular scale, or on the cell scale, or the tissue scale? And then some, but by far not all couplings were modeled in detail. I will show you that. But that's, again, when you're in physics, you understand this. It's impossible to model everything. You have to make it simpler and simpler. This slide is too busy. I will come back to this later. I'm not going to explain this right now. Sorry. What we then did was we created what we call a scale separation map. In some other projects, we have also been working on methods for multi-scale modeling applied to biomedicine. This is one method that we have developed. So I'm not going to speak about that too much. But what you see here on the horizontal axis is the temporal scale. So this is not time, but really a typical time constant for these processes. For instance, here you see cell cycle. Well, for the pigs, for the vascular smooth muscle cells, the cell cycle is 30 hours. And so that's order 1 day. And so we put it here. But we also have very fast processes that happen here on the level of microseconds. Well, here we have the spatial scale. And what we did is we basically named all the processes that we have, and we have organized them on this plot. So all these boxes have a clear meaning. This is all linked to the literature, and then we started to make all the connections and to see how these things are linked together. Blue is basically physics, and green is biology. That's how we call it. The interesting thing was, by doing this exercise, and this is all based on the literature study, some people who were already working a lot on instant restenosis started to see all kinds of interesting links and could already deduce lots of new insights from a simple picture like this. And so you could call this a qualitative model, qualitative multi-scale model. We applied the same idea to models for cancer, for tumor growth, and to some bladder disease and some other things. And it is insightful. It's an interesting exercise. We also tried this for to model thrombosis, and that was not so interesting because it basically all dropped on top of each other, and that was not very insightful. But in this case, well, we immediately realized if we want to build a mathematical model of this thing, this is way too complicated. So we simplified this, so we left out many processes, and we made it even simpler. And in the end, we started to implement this. And I will quickly talk you through it. So we have one big box here, which is called smooth muscle cell proliferation. So what we do there is every single smooth muscle cell in the arterial tissue we model as an agent, and we model the cell cycle in each cell, and we model the cell proliferation, so the fact that it grows and it divides. And that process we basically couple to two other important processes. One is blood flow, so blood flowing through the artery, which gives a shear stress. The shear stress is able to give, I will tell you later how, to signal the smooth muscle cells to stop growing. So there is a link between the shear stresses on the surface of the lumen exercised by the blood flow, and that goes back here. Well, if the cells start to grow, the lumen starts to change, obviously the blood flow will change, and there is a link between this box and this box. You also find diffusion here, which is this idea of drug-eluting state. And so this is the box where we simulate the diffusion of drugs into the tissue. Okay. And now this is a quite simple model. We have already added more boxes, so we started to put more detail into this, but it works. Now, again, if you would then say, let's now make a two-dimensional model and make it very abstract, and you would show this to the medical doctor in our case, he got lost here. He said, this doesn't look like my system anymore. I told him, listen, in physics, you know, we are very used to doing this. We first make the models as simple as possible, just to understand what is going on there. And next we will try to get the realism in and go back to maybe the full-blown system. Exactly, we went in that direction in this project. So we said, first let's do it in 2 dimensions, that we can understand. We can easily run our simulations, no problem. We learn a lot, and then we go back to the full-blown detailed 3-dimensional. The interesting thing was, so the cardiologist didn't believe it, but after the first results got in, he started to believe it. That was an interesting exercise. And we now got him to a point that, you know, he says, yeah, why don't you do this in 2 dimensions first? However, we also realize that if you do this and you show this to biologists or to other people in the field, in the end you have to do the full-blown 3D model. But I don't think it will bring me much more insight than what we already got through doing this exercise. So, what you're looking at here is our simplest model system, but we learned a lot. So, we replaced this completely complicated stent by this simple square thing here, which is just a strut. Remember when we made a cut through the system? Well, here you see arterial tissue, and is there a whole lot? Yes, there's a global. You can see it here. So each dot is basically a vascular smooth muscle cell. We allow them to grow, to proliferate, they can move. I'm not going to talk about the details of this model, but I can send you more details if you want. The little yellow things here is this internal elastic lamina, so it can be broken. And because of that, you know, the cells can actually get into the lumen. Well, at the same time, we model the blood flow, and the biology of the blood flow is proper through the shear stresses. Okay. Now, what did we do with that? So we measured the response, which is the development of the incident restenosis, as a function of the deployment depth, remember that's the injury score, as a function of the size of the strut and the shape of the strut. This is interesting, because when we started this project, we said that, you know, this is probably not too complicated, we want to do stent design. We want to design the medical device to optimize it so that you develop minimal incident restenosis. And we still believe that this is possible, but this will take a little bit more time. When we showed these 2-dimensional results to companies who actually made these stents, they were also immediately interested. And because for them, you know, making a stent is about the engineering, how do you actually create such a thing? And how do you do that in an optimal way? And how do you do that it has the right mechanical properties? But actually modeling the effect that the device has on the physiology itself, that was something, of course, they do lots of animal studies. But trying to do this, and then taking that in the design loop of the stent, that was a new idea. And again, we are still developing these ideas. Okay, I was at that time more worried about validating a model. We were putting lots of stuff together and it doesn't make any sense. And so, for me the most interesting result is, do we actually get a good validation? Can we compare in a qualitative way with the available data? Well, this is how it looks like. Later on I will show you 3-dimensional results. Yeah? So we didn't stop at the 2 dimensions. But I hope you can see it, but here we have 7 days, 14 days, 28 days, and here we have put in a very small strut, and here we put in a bigger strut, and we then analyze the growth of this thing. And so it's of course fun to look at the movies, and you can of these simulations, you can look at all details. What we did in the end was to create curves where, as a function of time, we measure the incident restenosis, and we measured it by measuring the total number of new cells. So I just counted the cell number, but this is related to the experimental numbers. Remember, in our experimental data, we only had reliable data at 14 days, so here, and here at 21 days and 28 days. So, you know, I am not claiming that our models are validated or whatever, because we just don't have enough data. So we also need more pigs to do experiments, by the way. That's another story. It was interesting. We always find these nice logistic curves for the growth. And so we basically said, let's, so you see an initial exponential increase in the number of cells. Well, you could say, okay, these vascular smooth muscle cells, they migrate into the lumen, they start to divide, nothing stops them, they don't push each other away or whatever, so it makes sense that initially you find an exponential increase, and then, you know, something stops them from growing, and they go to an endpoint. So we could say, okay, if it's really a logistic model, we only need two parameters to describe this curve, which is an initial growth speed R, which is this initial exponential increase, and a maximum number of cells, which is basically this plateau, and the rest is easy. But we could fit all our simulations with this simple equation, and then you can easily summarize the results like this. I'm sorry for the very small fonts, I'm not sure if you can read it, but as a function of the deployment depth, so as a function of the injury score, we have measured, first of all, this growth rate, which is this initial response, and we see that it nicely correlates. So as the deployment depth is deeper and deeper, larger and larger, the initial response is faster. This we also saw in the pig data, remember? And so we were quite happy to see that. By the way, also the timing is pretty good. So if you look at where the whole response silences, it's about 4 weeks. And we didn't put that in, so we just put in some basic parameters, and this is how it came out of the model. So that we were quite happy with that. The thing that is wrong is when you look at this endpoint. So we find here that for all cases the endpoint is the same. And that's because in the initial model we had a way too simple stopping condition. We basically said that these vascular smooth muscle cells, they stop by some process if the wall shear stress has a certain threshold. And I'm not going to talk you through all the details, but under that assumption it makes perfect sense that they all go to the same endpoint. We know that's wrong, so we had to put a little bit more detail in the model. What we already could do is to look at the effect of the shape of the stent, whether it's drug eluting or not, and because we have these nice logistic curves, then we can basically summarize everything in terms of only two parameters, the growth rate and the maximum number of cells. And then you can start to compare what we call a bare metal stent squared with a drug eluting stent, and we find that the growth rate went down, and even if we change the shape from square to round, we find that the growth rate goes down. These things are also reported in the clinical literature. This is known. And if the struts become larger or smaller, then we find effects that we also find in the clinical literature. Is this validated? By far it isn't. And we need more data. But we are convinced that everything that we know from the clinical literature and from the pigs experiment, we reproduce in a qualitative way. Except this endpoint. That's not good.
What is the physical explanation for the difference between square bar and round bar?
So that would be this one and this one. My feeling is that it has to do with the initial growth. So you just create more space for the cells to easily and quickly grow.
So the perimeter of square is larger than the perimeter of...
Yeah, that's, I think in the end, if you would, so we haven't really looked at it in detail, but if you would look at this perimeter, or if you would look at the amount of metal that you actually put into the system, it will correlate, for sure.
Exactly.
And this is known from the literature. So if you make the strut smaller and smaller, you get less instant restenosis. Because, and that is a new insight, this growth rate goes down. Because as I will show you, this endpoint is determined by another process which is independent of what you do with the stent.
So maybe one question. If
Sure.
you have different scales of your vessel,
Yeah.
is it possible to make some scaling law of the growth for
I think
different
so.
scales of the vessel?
So let me tell you a little bit. So one assumption that we have made in the model, which is important, is that you should ask yourself the question, how much blood is actually going through this system? And we made the assumption that as the stenosis develops, that the total flux of blood stays constant. Why? Because the heart muscle needs blood, so the heart will basically start pumping hard. This is a very reasonable assumption. We know that this happens if stenosis develops. Either the microvasculature can open up a little bit so that the resistance goes down and you get it's possible to do that, or the pressure gradient over the stenosis just increases to maintain this constant flux. If you talk to cardiologists, they will also say yes, this happens. This is a reasonable assumption. There's one thing that is problematic, and these are the so-called collaterals. I don't want to talk about that, but these are basically new vessels that can form. Okay. But under that assumption, and if you assume that this is just simple Poisson flow, you know that with a constant flux, if the diameter decreases and decreases, the wall shear stress goes on. And there is always a same diameter at which the wall shear stress reaches the threshold, and then everything stops. So that's the reason that we always find the same incident restenosis as an endpoint. And that's also exactly the scale you mean, because it's really a matter of a diameter or a size of the vessel and the flux and the threshold. It's a very simple relationship. And in 2 dimensions it's a little bit different than in 3 dimensions, but then you can write down the equations yourself. And so obviously we miss an important process here. And that's, maybe just one graph. Well, I just discussed this basically. So this is what we see in our simulations. These are the data that we got from stenting the pigs. And qualitatively, the time scales are right. This initial response is right as a function of the incidence score, but the endpoint is not. So what we did is we went back and we started to look in a little bit more detail at the biology. And we said that, and we already knew that a very important process is the so-called re-endothelialization. So you need new endothelial cells in your artery, and they will in the end silence the vascular smooth muscle cells from growing. So what happens is here you have the vascular smooth muscle cells. They are proliferating, and they will only stop proliferating if they get the right biochemical signal. And there's one pathway by which they stop, and that is through nitric oxide. If there is a sufficient level of nitric oxide in the cells here, they will go back to their normal state. They will stop proliferating. And the cells that create the nitric oxide are the endothelial cells. So you need to have a healthy endothelium inside your artery, and then once it's there, they will start to develop or produce nitric oxide, and that will only happen if the shear stress is in the right range, in a physiological range. And we know that if the shear stress is high, the low endothelial cells look pretty strange, and they don't do what they cannot do this regulation of the vascular smooth muscle cells. So we now see another process occurring. As I said, we know that after the balloon angioplasty, all the endothelial cells are gone, so they have to come from somewhere. Well, we had to make a few more assumptions, but basically what we have added now is a model for this appearance of the endothelium, and then the reaction of the shear stress on the endothelium and the NO production. Again, I'm not going to go through all the details, I will just show you the final results. It's a pretty technical story, but it's all published by the way, so you see, let's just accept it here, but it's published by now. We basically studied 3 case studies of the appearance of the endothelium. And I'm not going to talk you through the details again, but we have 3 scenarios based on literature, where one scenario states that the endothelium reappears pretty quickly, in a matter of a week, 2 weeks. And another scenario says, well, you need at least 3 weeks before you have a fully functional endothelium. This is again based on extensively reading the literature. And this is also the best that we got. So if we want to really validate these kind of models, we just have to do the experiments ourselves. Or go back to the pigs and look much more carefully what is going on. Or there's some other experiments. We have been thinking of harvesting the coronary arteries. So friends, colleagues of mine in Eindhoven University, they go to the butcher house. They take pig hearts from the butcher house. They take out the coronary arteries, bring them back alive, and then start to do experiments with them. We are currently setting up, so you don't have to have live animals anymore. These hearts you basically get for free. So if we are able to do that, we can put all kinds of equipment around it and just see what happens. The cardiologists don't believe that anymore, that kinds of ex vivo stuff, but it allows us to do lots of detailed experiments. We are setting it up right now. Now, what do we see? Let me quickly go to the results. So here again we see our growth curves. So this is our original model, remember? So as a function of how deep we push, we see this positive response, positive correlation, but in the end they all go to the same endpoint. Now, here we have the three cases with the re-endothelialization. So again, initially we see a positive correlation between injury score and the speed of the response, but now we find different endpoints. And this is because it just takes a more or less constant amount of time for the endothelium to recover. And if the initial response is faster, of course you go to a higher endpoint. It's very simple. So if you would write this down in terms of rate equations or a simple ODE system, I'm sure you can come up with a model that will reproduce this completely, and you just have to put in the right physiological numbers, and you're in business. And well, that's going to be one of the next steps. But what is also interesting is we have these three cases, and I'll note a little dotted line here. So what we have done is to say a cardiologist would consider the response a pathological response, so real instant restenosis, if more than 50% of the lumen is covered again. And that's exactly this dotted line. So basically we now have a scenario, or let's say a model parameter, that will dictate whether you develop instant restenosis or not. We know that with bare metal stents, only 10%, 10-15% of patients develop, and the rest do not develop. It would be interesting to know, before you receive treatment, whether you will develop it or not. Because then you can either go in with a bare metal stent or a drug-eluting stent or whatever, do something else. Now we say that if we would be able to measure this re-endothelialization rate, or so is one way or the other get our hands on this parameter, we have something interesting. Of course, we assume that the re-endothelialization is the only thing that determines where you go. There are other parameters. I already mentioned the collaterals. We're looking into that. We know, for instance, that sometimes you have genetically, or sometimes you have a genetic defect by which you have slow wound healing, and by which you have not so many progenitor cells in your blood. And we know that these people tend to develop more instant restenosis. That is one of those correlations that was found in the clinical literature. So that would, tell me that they would be here in this case. So it takes more time, so you develop it. Okay, so this is what we got out of the 2D model. We went even more on, and we said, where do these endothelial cells come from? Is this like there are progenitor cells circulating in the blood, they home in on the tissue, so you get what we call a random endothelial cell seeding? Or is it like that they develop from outside, where you still have endothelium, where the balloon didn't take away all these cells, or will they grow inwards in the vessel? If you would run these two scenarios in the model, and that's easy to understand, if you would have random endothelial seeding, you would expect a nice, well, more or less flat surface. If you would have endothelial cells from the sides growing in, you would expect more neointima, inside the system, so halfway the stent. Well, that is something we can still test. So we went back to our histology, and what we actually did is, if you look at the way this histological database is built, and so you take a stented artery out of the pig, it is frozen, it is sectioned, and then we look at the sections. And so we basically have, for a number of stents, we have them nicely numbered, and we know exactly where the cross-section came from. Well, for each cross-section, we then measure the lumen diameter here we measure the total diameter, and the difference of the two is the incident restenosis area. And then we can plot that as a function of position. Now we don't know exactly the length, that was not kept, but we do know the position. So we can actually see if we find, you know, either this scenario where it looks more or less flat, or where you see a clear, you know, increase in the number of new intima as you go through the step. And this is interesting because there is an ongoing debate where these endothelial cells actually come from. Okay, well, we did the experiments, and if you look at all the arteries that we saw here, you have 1, 2, 3, 4, 5, 6. So these are eight standard arteries that we did. It is a little bit messy. So on average, it looks like that you have pretty flat curves, but you also have some outliers. And basically, what we did is to, you know, just take the average of all this, and then you find this curve, which is more or less flat, but there is something more going on that we still don't understand. But you know, we believe that we have, and there is much more detailed in this study. I'm not going to discuss that. We have probably excluded this effect, that the stents grow in from outside. So there must be some random seeding of the endothelial cells. Does this mean that these are progenitor cells? Some people don't believe that they exist, that they are kind of, you know, not the pluripotent stem cells, but something that can at least develop into endothelial cells if they would come into these sites. Well, when we started this study, there were some biologists in Sheffield that said, yeah, these cells exist, and now they say, no, that's bones. So I don't know. Another option could be that the endothelium is not completely removed, but you have patches of cells, you know, after the balloon angioplasty.
That was exactly my question.
Exactly, there could be patches of cells just sticking there.
Why it is removed completely, destroyed? Why?
Because people have made, so I have seen a number of studies with EM, electron microscopy, where immediately after treatment the arteries were taken out. And if you look at these pictures, there's no anything. You have to realize that you go in with a balloon, then there's an enormous pressure put on the balloon, you know, and it's moving. So all these cells, many of the cells are gone. That we know. But it is possible that not everything is gone. And that's probably what is going on there. But that is something we would like to better understand. But and that endothelium is gone after this treatment, that we know. Yeah, but probably not all. I will show you the pictures if you find this interesting. But it is something that we believe is true.
Well, it is quite interesting, truly speaking, because first of all, yes, first idea that there is no stem cell or any undeveloped cells in the bloodstream.
Exactly.
If you find them, it means something wrong.
Yeah.
The other belief, not belief, but it's all proven that pericytes are the actual progenitor cells that are going. But the question is if they are going inside, because usually the theory is that endothelial are just coming according to hemotaxis, they are coming and close the defect.
Exactly. And you know, I mean, if it would be those cells circulating in the bloodstream, you could actually, you know, try to measure
it. Happens.
But I mean, but that's not what it is. So we believe now that it must be, you know, and probably it's a combination of both. So of course you have healthy endothelium, you know, up and downstream, so they could slowly grow in. But then we should see this disappearance that we don't see in the histology. But we do see this effect. So I mean, this is downstream, this is upstream. So something is happening there, and we still have to find out. Well, the only way to find out, this is where our modeling stops. We have been able to interrogate the system a lot. We have been asking some pretty straightforward questions. This is what we find. Now we have to go back to the experiments. We have to go back to the pigs or to the mouse and see what we're doing.
Am I right that if such a stent would exist, which does not destroy the tendon during implanting, then the problem of restenosis will be solved?
That's what this at least suggests. if the endothelium, so we assume, so one question that we haven't discussed yet is why would these smooth muscle cells start to proliferate? And we believe that this is because you put such an enormous pressure on these cells. Again, you have to realize you come in with a balloon, you put 10 bar of water pressure on this. So it's a huge kick that you give to all these cells. We believe that because of that, they go from their standard contractile state to a synthetic state. And now the question is, how do you bring them back to the contractile state? Now we believe that most of the cells are just, they're inside the tissue, so that there is contact inhibition or whatever. They quickly silence. But there are cells that are capable to get into the lumen, and that's where they can freely start to proliferate whatever. So if you damage one way or the other the internal elastic lamina, and we believe that is what happens due to the struts, that gives the cells that are close to the surface the option, the possibility to migrate into the lumen, and that's where they actually proliferate. So even with intact endothelium, the problem is not gone because you have these smooth muscle cells that get into the lumen, and that's where they start to develop basically the scar tissue. And there's more going on because there are growth factors, there is some thrombosis, there's some other stuff. So lots of the biochemistry is just completely, that's the fun part, but also the danger of these kinds of mathematical models. You just skip all the details. And sometimes you can do that. Okay, I told you before that this was also done in 2 dimensions. We also do this in 3 dimensions. And this is something that a cardiologist will start to recognize. So, I mean, it's hard to see, but if you look carefully, here we see a cross-section of a stented 3D artery. Again, we do single cell simulation, so every little dot you see here is basically a smooth muscle cell, and here you see the initial state. For comparison, here you see an electron microscope picture of a stented coronary pig artery, and so the way this stent is pushed into the tissue, again, we believe that this is done in the right way, and we see this nice growth, and at some point it stops. Now, we are still looking at in detail at these results. So we again get comparable growth curve as we had in the 2D model. They look different, of course. They should. I mean, you have a degree of freedom more, and lots of stuff is happening. You don't see error bars on these curves yet, so these are the results coming in right now. They don't silence early enough, so I mean, there's still many things that we don't understand, but we are quickly getting there. This is also the part where computer science and scientific visualization is important. So here you see a guy operating a huge iPad, a big touchscreen, where you can play with his data and just look at it. And if you can't look into a microscope, you need to have something nice to convince you that people will give you money and do something interesting. But I think that in the end, we are developing also multi-scale visualizations that allow you to go deep in a cell, look what's going on there, and then zoom back to the full picture. Now, we are also doing this, so this is a much longer vessel. This is interesting. This would be, a length of a real strut, of a real stent. So we can do the full stent now. We still have to increase the diameter of this vessel by a factor of 2, and then we are really doing coronary arteries all the way. And it's just a computational problem. You need big computers to do this. Again, I don't want to discuss that, but this is work in progress. But in a year from now, we will have this up and running, and then we can do a real-size stent in a real artery, and that is something the biologists and the cardiologists will do. And then we will make it curved, and we can finally get to a point that we can do a much stronger validation of this model. We have some very good human data and big data for this full-blown system. And once we have that, then we get to a point that the model is validated and we can actually start the predictions with it. Okay. I will bother you a few more minutes with another part that we haven't discussed. Just quickly to show you again where I believe you can go with this type of modeling, and where you can try to close the loop with the experimental data and make it an iterative process. And we talked about the endothelialization, where I believe that it would be so nice to get some experiments now where we can look at this in more detail, because if the way we look at this process is really true. It's basically two processes, one fast process, one slow process, and the slow process in the end determines the amount of incident restenosis. So if you can get the rates of the slow process, you have a very simple parameter in the end that will determine what the treatment does. And then you can go back to the stent developers and start to play with it. Okay, and so we want to do that kind of modeling. And so here is a final attempt where we look at this first phase of the incident restenosis. So I told you, as you push the stroke deeper in the tissue, you get more response. But the question is why? We also found that even if you push the stroke a little bit into the tissue without damaging the internal elastic lamina, you still get an incident restenosis. And again, the question is why? So we developed another hypothesis that we wanted to test in a model. I'm not going to read you through this whole thing, but basically what the model says is that if you look at this internal elastic lamina, it is not a rubber sheet. You know, it is also a layer of tissue which has little holes in it, fenestra. However, it is an elastic thing, we know that. So our idea was, if you push the stroke deeper and deeper, you make the holes larger. And then it becomes easier for smooth muscle cells to migrate through these holes into the lumen. So it might be that if you push deeper without damaging, you make it easier for smooth muscle cells to get into the lumen. The more you get into the lumen in the first day of the response, the quicker you see an initial response. Well, we know that smooth muscle cells can migrate through these holes in the internal elastic lamina, that we knew from the literature. But to find this connection between the initial rate of the restenosis and such a simple, let's say, mechanical effect, that was not known in the literature. So we decided to develop a simple model that could maybe, let's say, how to put it, to give some meaning and some realism to this hypothesis. And again, if the model would be positive, then that could be a reason to go to the lab and actually test it. Well, we did that, and again, for now, here we developed another model, which is called the cellular pots model. That model allows you to put a little bit more biological details into the model. So we now use the cellular pots model for a model of the arterial tissue, and we don't do blood flow here, so nothing stops it. It's just starting of the model. I don't know if you can see it. Here you can see this yellow stuff here. That's the internal elastic lamina. It now has holes in it. If you push deeper, the cells start to migrate. And here you see a nice movie of the developing cells and the developing incident restenosis. Well, let me quickly go to the results of what we have done. What you find here now, as a function of time, you find to know, first of all, the number of migrated smooth muscle cells. And these curves are again for different injury scores. So what happens is that if you go from a small injury score to one that is a little bit larger, you indeed find that you get more cells that migrate into the lumen. If you damage the internal elastic lamina, so that would be this curve, and you get much more cells into the lumen. And then after 1 or 2 days, so the whole cell cycle kicks in, they start to proliferate, and you get this exponential increase. And that's what you see here. This is where we actually turned on the proliferation, and you find these curves that we already observed in the original model and also in the big data. And so we believe that this is one hypothesis that would lead to this initial response. Again, we have to test this. Well, there's not so much more we can do now, and we are setting up, as I told you, with these ex vivo experiments, taking pig hearts from the slaughterhouse, and we are setting up experiments to actually observe this initial behavior after a few hours. But of course, well, we don't know what this will bring us. So what have we learned? We first of all set up this multi-scale model for incident restenosis. I didn't talk at all about the details of this mathematical model. I just talked about what we did with it. But actually developing such a model already brought lots of insight. It's all based on reading the biological, biomedical, and biophysical literature, and bringing everything together. We are capable to reproduce the in vivo porcine data, which is rewarding, it's good, but I'm still not claiming that the model is validated, the mathematical model. So to actually apply this instead of design or whatever, or to do predictions, we're not yet at this point. We need more detailed data. However, we did develop deeper insight into the disease. So what is actually going on here? And you know, you could claim that lots of this was already in the literature, but we brought everything together and we saw the whole thing in the context of the overall process. I think that's where the power of this mathematical modeling is. I think that is one way where computational biomedicine can help biomedicine or the medical profession. Well, we formulated a number of hypotheses. This re-endothelialization, that's one, and the second is initial response. And we are now basically trying to get money to do this test in the lab. So how should we proceed? What we are planning to do is, of course, these 3D simulations. That will go on. Once we have realistic geometries and we can include curvature, we have one data set waiting for us that is extremely detailed, and that will allow, if we can run our 3D simulations and get a good comparison with that data set, I will claim that the model is validated. So that is going to be an exciting year. We want to get more data in, as I discussed, and then, you know, we want to study STEM designs. That's going to be definitely the next step. Once we have done this, can we now improve the treatment? Or can we personalize the treatment? But then I don't believe it. It's probably going to be on the level of population studies. And well, we have to discuss that in more detail. But still, one thing that we're also doing is the models are far from complete. So we would like to get more detail into the models, but we only do that after we got more experimental data. Of course, we can, I mean, you can go on forever putting in more and more details in your mathematical model. Does it make any sense? It's interesting to do it. It's a nice master thesis project for sure, but it will not give us new insights into what we already know. But things that we would like to do is to look at the role of these collaterals, the smooth muscle cell biology, extracellular matrix, pulsating transmural pressures, all details that, you know, remember this first scale separation map where we had all the green boxes, all the stuff we kicked out basically, we want to bring in back if the experimental data tells us to do so. Okay, these are just some projects that funded all this work. And this is, of course, the most important slide. These are the people that actually did all this work. You know, that's and it's not everybody, but there were basically 3 PhD students down here and 2 postdocs who put lots of time in developing the models, running the models, comparing the data, etc., etc. So it was a big team of people. I think I will stop here. I took more than enough time for me. So thank you for listening to this. And again, if you want to have more detail, let me know. I can send you all the papers and everything we have.
Thank you. Thank you.
You're welcome. Really. You're welcome. And again, this is really an example of where I believe computational modeling can go in this field of biomedicine. Sure, please.
Previous slide. No, it's on.
This one.
No, one more.
About the money. This one.
Last phrase. Biology to pulsating transmural pressures.
What I mean there is the following. We know that that the vascular smooth muscle cells do not react to shear stresses. And that's why I brought in the endothelium. We do know, at least that's what the literature suggests, that vascular smooth muscle cells react to transmural pressures. And so the transmural fluid that goes through the endothelium and is perfusing the arterial tissue, you have some pulsating pressure in there. And we know that if that is outside and we know this, by the way, from tissue engineering studies, if that is outside the normal range, the smooth muscle cells will start to proliferate as well. So they will become synthetic. And we want to bring in that effect because we also believe, and that's part of the literature that we haven't really studied very good, but if the pulsating pressure, therefore, is not back to its normal range, the cells will not silence. So there could be another mechanism by which they will continue to proliferate, even if there is sufficient nitric oxide in the system. But, you know, this one came by discussions with one of the biologists in Sheffield who keeps repeating, you know, you have to do something about this transmural pressures. We see this in tissue engineering, we see this in other places. And she believes that this is a very important process. So, it's a matter of reading much more about the specific biology of these smooth muscle cells and see if it would make any sense to bring that into the model. And you know, I repeat myself, but I first need more experimental data to do that. I don't believe that it will bring us much more right now. But the interesting thing is that, you know, even if you want to understand all the details of how a artery reacts to, you know, just the standard pulsating blood flow and everything that happens in there, that's already extremely complicated.
Normally you do this at the time.
Sorry?
There are mathematical models of pulsation processes.
Yeah.
Does anybody do this?
Oh yes, yeah. So, I mean, doing...
And mathematical models of such process.
Yeah, so it's basically solving the Stokes process, right? It's, so that, I mean, if you look at state-of-the-art computational hemodynamics, what we can do in curved vessel, do full-blown pulsatile flow, fully resolved, also fluid structure interaction. So that's basically all under control.
Wait, but you considered steady.
We did steady flows.
And you motivated this consideration by saying that the flux is constant and correlated.
Well, so
this is known by doctors.
There are two things. One is, first of all, we did steady flows. It was very simple. Computationally, it would become too heavy to do pulsatile flow at the point in time that we implemented this model. So we said we do steady flow as a kind of surrogate, a kind of averaged, as a kind of average signal, because we know endothelial cells react to oscillatory shear index, so that's time averaged shear stresses. But it's you know, that's is it here? I think it's here. Well, I mean, it should have been here as work in progress. Of course, we also want to run the 2D simulations with full-blown pulsatile flow to see if it makes very much difference for the studies that we have done with static flows. I don't think so. If you average over the signal, it will be in the details. But the other one, so your remark about the steady flux, that's a more important one. And so the whole idea is, as I told you, the physiology. So if you develop a stenosis, what we know is in patients, if you develop a stenosis, typically your physiology tries to compensate for that in two ways. One is, no, in three ways. One is by reducing the resistance of the upstream vascular bed. So, and that can do by dilating, so by just making the microvessels a little bit larger.
Not upstream, downstream.
Sorry, downstream, of course. Thank you. It's getting late, I'm getting tired. So downstream, that's one. The second is by increasing the pressure gradient, so basically the heart starts to pump more. And the third mechanism are these collaterals. So these are, you know, arteries that develop, you know, as part of the physiology to help the system to fight this bad situation of the stenosis. You know, the interesting thing is, that's something we started to realize and we start to take into account. If people have lots of collaterals and you develop a stenosis, that's bad for restenosis, because basically if you develop a restenosis, the collaterals take over part of the blood flow. Therefore, the shear stresses in the stenos part don't become high enough, and you don't get enough NO production, so you develop more incident restenosis. So having a mechanism that helps you when you develop your original stenosis is bad to save you against the restenosis. And so I've had discussions with the cardiologist who said, yeah, well, and so we basically should block the collaterals then. It's a crazy thought, and he was not really serious. But, you know, the bottom line there is that physiology is so extremely complicated. If you think you take everything into account, there will be yet another process and yet another feedback loop that does this. But this whole assumption of a constant flux, we now basically understand what it means and what it does to the system. And if it's not if the flux goes down as you develop a restenosis because of the collaterals, or for some other reasons, we know what the response will be. You know, I liked your original remark about can you come up with simple scaling laws? Yes, you can. And in the end, what we would like to have is to have, you know, a few parameters that will actually determine the amount of restenosis that you develop by running many of these simulations. I think that's where we're going in the end. Very, very simple, almost engineering solutions where you just have, you know, a very simple equation where you put in all the details of the physiology. Let's see. But
You were placing stents into the healthy efforts.
Yes, that's another good remark, of course, you know. So we are stenting perfectly healthy animals. So what happens if you have an artery with lots of mesh in there? Well,
it's already compromised.
We have lots of, let's put it like this. We have some human data sets, and that's going to be a next step. So we have some data where before, and that's really a stenosed artery, obviously. Yeah, these were real patients. What happened is that before the treatment, this was done in a medical center in Rotterdam, they went in with OCT probes, so they could, and IVUS, so they could do a complete imaging of the room, including the plug. You can see the plug very nicely. Then the stent went in. They also have X-ray images, and so we know the complete geometry, and we have follow-up studies. So we know what happened. And so these patients came back, and we did another IVUS on these patients. So we have the full-blown data set. Unfortunately, it is the resolution is very low. So at that time with the IVUS, we were quite happy to get some results. But this is a data set waiting for us where we actually have diseased arteries. So one thing that if you think about it, if you have a diseased artery, you would expect a restenosis on the part that was still healthy. Because, you know, the part that was not healthy, there's so much material in between, no way that you will get something growing there.
If it's not intrinsic.
We don't know. You see, but we are currently working with a group in Cyprus to develop mouse models and rabbit models, where you first, develop plaques and then you can actually treat them in the aortas. So that's where we have to go back to the animal models again.
Because metabolism is different there.
Yes.
The way cells behave is different.
Exactly. We know. So it's. But I think that, it would be where we want to go with this work, but with any other modeling work that we do, it has to go hand in hand with biological labs, where you can do very detailed studies, and it has to go hand in hand with studies on human patients, so really clinical studies. And sometimes you're lucky you can do that. So with the context we have in Sheffield, we can actually get close to, we have good clinical data, we have good clinical data from Erasmus, but sometimes the processes are just too complicated.
The only way to solve it, that's American do in Russia, I think they don't do it because it's forbidden by law, but there are heart donors, and sometimes the heart is not good enough to be donated, and there these hearts are sort of still alive, used for research science. That's where you can take the artery and try to stand in there, and you can keep it alive for like at least 72 hours and to see what happens there. That would be the way.
That could be interesting. Yeah, so another thing is that you can actually, you can take pig hearts out of the pig and keep it alive for at least 2 days.
Yeah, but you need to compromise that way.
Exactly. I don't think there are good pig models that develop stenosis. That's the problem. You have to give them lots of whiskey and lots of greasy stuff to eat, and then maybe it will. I don't know. Okay. we do this kind of modeling a lot also in the intercranial aneurysms, also in modeling tumors or cancer growth. We have done this in HIV, so the whole, basically the whole process that leads to an HIV infection and the details of the RNA, of the virus getting into the cell, but also on all the other levels. So how the virus then spreads in the body and from person to person, etc., etc. And as always, we try to be as close to real data as possible. Otherwise, it's going to be artificial. It's fun to put that in a computer, but you don't learn so much. So that's wonderful. Okay, that's it for now. Thank you.
One more question.